disease called neuroblastoma, and today I'll be giving a brief update on our efforts to discover the genetic basis of the disease. Many people initially think that neuroblastoma is a brain tumor, but it is not. It's a malignancy of the developing sympathetic nervous system. Children can develop tumors anywhere along the autonomic nervous system. Oftentimes they develop in the adrenal gland. Many kids present with metastatic disease. It could infiltrate the liver. There are often bone marrow metastases, and we sometimes see paraspinal tumors as well. So it's the most common malignancy diagnosed in infancy under 1 to 2 years of age. However, it's overall a very rare cancer with only 600 to 700 new cases diagnosed in the United States per year. The median age at diagnosis is only 17 months, and it's a fascinating disease in that it has a very diverse clinical presentation where a subset of tumors may undergo spontaneous regression. And indeed, there's a subset of neuroblastoma called 4S that actually has the highest rate of spontaneous regression of any known human tumor. Yet we also see patients on the complete opposite end of the spectrum that have very aggressive and refractory disease. The phenotype of these tumors can be defined by the genomic alterations that are present in the tumor. Most notably, there's often amplification of the MYCN oncogene. This occurs in about 25% of neuroblastomas. And then we also see these segmental chromosomal copy number alterations in the tumor. And we also know, which I'll present some data in a moment, that there's a significant contribution of germline variants to tumor phenotype as well. And the Children's Oncology Group has classified patients into low, intermediate, and high risk of disease. And in addition, there's also an international neuroblastoma risk group assessment that includes very low risk. And if you look on the right, what we know is that those tumors with segmental chromosomal aberrations are typically very aggressive, and they're considered high-risk disease. Whereas those that don't show these segmental aberrations and may have only whole chromosome gains or losses are more benign tumors and are classified as low. And then there's a less well-defined subset of intermediate tumors that fall in between. And these risk groups are used for treatment stratification purposes. When we look at the Center for Disease Control at the number of childhood deaths by disease per year, we see there's over 4,000, and the majority, 53% here, come from childhood cancer. And I mentioned that neuroblastoma is a very rare cancer overall, yet it accounts for a disproportionate number of these cancer deaths, in estimating 12% overall. And those that do survive high-risk treatment often have significant treatment-related morbidities. So we're interested in studying the genetics of this disease because understanding these underlying genetic factors that influence tumorigenesis will help us inform rational therapies and is really the first step towards understanding or preventing cancer. So we know that only 1 to 2% of cases present with a family history of the disease. These are typically children that are diagnosed at an early age of onset. They may have multiple primary tumors, and it's been particularly difficult because multi-generational pedigrees are rare due to the potential lethality at a young age. And if we look at the right, we see that back in 2008, the major neuroblastoma predisposition gene for familial cases was identified as ALK through a traditional sort of linkage study. We also know that another 5 to 10% of these familial cases have mutations in FOX2B. And these are really patients that have other associated conditions of neural crest-derived tissues and are considered syndromic. So this doesn't account for all families, so it's thought that there's other familial neuroblastoma predisposition genes to be identified. And we also know that even patients that do have an ALK or FOX2B mutation, that there's incomplete penetrance. So just having these mutations does not necessarily mean you will show up with a disease. It could be that some of these tumors underwent spontaneous regression. They could have clinically occult tumors that we don't know. The primary was neuroblastoma. And the search is really ongoing to identify modifiers of these known highly penetrant mutations, as well as identifying additional familial predisposition genes. But as I mentioned, this is only 1 to 2% of cases. And the question that we've really been trying to address in our lab is, why does neuroblastoma occur in children without a family history of the disease, which we refer to as sporadic neuroblastoma? So this is a graphic from a recent review we published, and how we tend to think about the variants and their germline variants and their contribution to susceptibility. So along the x-axis, we're plotting the allele frequency, where we start with the very rare alleles, and on the right, we're showing more common alleles. And on the y-axis, we have the effect size. So the further up you go, the bigger the effect size. And if we look up top, these are the known familial neuroblastoma predisposition genes I mentioned, and the rest of the genes here are really focused on studying sporadic neuroblastoma. And we divide these based on whether they're common variants. These are alleles that are present in healthy individuals, but the alleles are more frequent in neuroblastoma. They're enriched. And these have very slight elevated risk for developing the disease. Certainly, if you have one of these common variants, one is not enough to push you over a threshold to develop the disease. However, we do know that they contribute to predisposition. And these are identified by a genome-wide association study comparing large number of neuroblastoma cases to controls. In this case, these are children without cancer. In addition to the common variants identified by GWAS, we use sequencing-based approaches to look at rare variants. And these are alleles that are either not present in control populations, or they exist at very low frequency. It's been a challenge to assess the relative risk of these rare variants, because they require much larger sample sizes. However, we presume that they will have a much larger effect size. And as I mentioned, it's a challenge to study because we need large cohorts. So I wanted to first walk through, there are 2 large studies that we've been involved in that, as Adam mentioned, we're trying to integrate these data in with the Kids First data that we're just processing. So the first is a genome-wide association study. And these are cases and controls that we have genotyped using Illumina single nucleotide polymorphism arrays. We've genotyped over 6,700 neuroblastoma cases. These are all recruited through the Children's Oncology Group Biology Study. And then through the Center for Applied Genomics here at the Children's Hospital of Philadelphia, we have access to a little over 35,000 control children. These are children without a history of cancer, as well as neurodevelopmental disorders. So I'm just showing over on the right the clinical and biological characteristics of the neuroblastoma cases. And I put this here just to note that these are really reflective of the patient population overall, and therefore we think are very representative of the disease. Just to note, they're from multiple ethnic backgrounds. However, the majority of the individuals recruited through the COG are of European ancestry. So we take these data and we use algorithms to impute genotypes genome-wide, looking at over 3 million variants across the genome, and then basically test for allelic enrichment in cases versus controls. And if we do this, this is a Manhattan plot where each of these dots represents a SNP, or a single nucleotide polymorphism, that is either imputed or present on the array. We have the chromosomes listed in order here. And on the y-axis, we're plotting the significance as negative log 10 of the p-value. So we're really interested in these peaks here. And what we noticed throughout the years is that a subset of the loci that we identify are enriched in the high-risk form of neuroblastoma. And then on the flip side, we see other genetic variants that are enriched only in the low-risk subset of neuroblastoma. And then there's a group that really shows no bias. So we know it's still one disease. However, there is clear evidence from the underlying germline genetics, even at common variants, that there are 2 subtypes, one being high risk and one being low risk. I don't have time to go into it today, but we've also demonstrated for many of these that the causal genes that we think are at each of these loci contribute not only to initiation of the disease, but are really very important to maintenance of the malignant phenotype. So they either promote cell growth, promote metastasis, they really help drive and maintain the tumor. And that was sort of an unexpected result initially. So currently we've genotyped 6,702 neuroblastoma cases. This is based on blood DNA and over 35,000 controls. And I'll come back to that a little bit later. So in parallel to these efforts, we've also utilized sequencing data from the Target project that Allison mentioned a little bit earlier. So here we're looking at the neuroblastoma cohort of Target. This includes 222 exomes, 135 whole genomes, and we've looked at 499 targeted capture patients. And this study I'm going to mention here is based on germline blood DNA only. So we've looked at 758 unique patients. And unlike the GWAS data, this cohort is really enriched in the high-risk form of the disease. That was done through an effort to enrich for poor outcome when we studied individuals through the Target project. So what we did was call germline variants across these different sequencing modalities, and then pathogenicity was assessed according to the American College of Medical Genetics and Genomics guidelines. And I'm just going to give you a flavor for what we've seen. These are the genes that had pathogenic mutations or rare variants across multiple cases. And you can see at the bottom, they are predominantly in high-risk patients, although the cohort was also predominantly high risk. And we do see some in intermediate risk as well as low-risk patients. And the most frequently mutated gene was BARD1. And if we look a little more closely at that, we saw that there were 9 pathogenic mutations in the BARD1 gene across our cohort of 758 individuals. That corresponds to a little under 1%. Most of these were stop gain mutations. We also saw 2 splice site mutations and a frame shift deletion. When DNA was available, we validated all of these by Sanger sequencing as indicated. And we then tested this for enrichment against the exact cohort, excluding TCGA individuals. This was highly significant with a p-value of 10 to the minus 6 and an odds ratio of 14.99. This was done across all ethnicities, so we also restricted to European ancestry, and this remains significant as well. So this is sort of a flavor of what we're hoping to do with the sequencing data, and gives you an idea of the sheer number of patients that will be needed. So we have a longstanding interest in the lab of looking at copy number variants. And just so everyone's on the same page, these are segments of DNA in the genome that are either deleted or duplicated such that there's no longer 2 normal copies. So if we look on the left, we see this is the normal situation where we have 2 copies. One is listed, indicated in orange, one in purple. The individual on the left has lost the region in purple and therefore has a deletion. The individual on the right has gained that region and therefore has a duplication. So our hypothesis was that if we study large, rare copy number variants, that these will contribute to neuroblastoma predisposition with a much larger effect size than the common variants that we had identified through the neuroblastoma GWAS previously. And our approach was to test these rare germline variants, CNVs, genome-wide for enrichment in cases relative to control population. This, in this sense, being children without cancer or neurodevelopmental related phenotypes. And by rare, we were referring to any variant with a frequency less than 1% in the general population. And by large, we mean anything greater than 500 kb. And this work was really led by Laura Egoff, who's a very talented graduate student in the lab. So I just want to remind everyone who doesn't think about these things all the time how we can get this data using SNP arrays from our GWAS. So we're given raw SNP array intensity data with allele A indicated in red and allele B here in blue. So if we look across a panel, these individuals would be homozygous for the A allele, these would be homozygous for the B allele, and the purple dots here represent a heterozygous SNP. And from these data, we can get two key items. One is the log-R ratio, which can be thought of as the ratio of the observed intensity over a reference panel of individuals. And we can also get a B allele frequency, which can be roughly thought of as the intensity of the B allele over the intensity of the A and the B allele. So if we look at these plotted out where each of these dots is a SNP, we expect for a normal copy number, the log-R ratio to be centered around 0. And for the B allele frequency, we expect to see this spread. So some SNPs will be homozygous for the B allele, some will be heterozygous and have AB, and some will be homozygous for the A allele. When we have altered copy number, this changes. So in a deletion case, the log-R ratio would be decreased, and we would see homozygosity either for the B allele or the A allele in the B allele frequency. Similarly, we can detect duplications with an elevated log-R ratio and a split in the B allele frequency where we have multiple genotypes possible with 3 Bs, ABB, AAB, or homozygous for A. And then we apply algorithms and we can get copy number calls across the entire genome. We use the method referred to as circular binary segmentation. So this is a rather complicated slide, but I presented here to show you that the resulting neuroblastoma cohort was very well balanced across the clinical phenotypes, just as our input data was prior to extensive QC, which I won't go into great detail of. But we also did not see a large difference between, or significant difference between the discovery cohort after quality control and the replication cohort. So both were fairly representative of the patient population. In the end, we defined a discovery cohort of 3,309 cases and 8,855 controls, and a replication cohort of 2,276 cases and 14,650 controls. As I mentioned before, this is there are multiple ethnicities captured in this data, both in the cases and in the controls. So if we can take the genotype data and perform a principal component analysis and plot PC1 versus PC2 along with the various HapMap populations, we can very easily separate out Europeans, those with African admixture. We can identify East Asians and South Asian and Hispanic. We can do this in the discovery cohort and also the replication cohort. If we dig a little deeper and compare PC2 to 3, we can separate out the South Asian population with those with a more Hispanic contribution. And again, if we look at the discovery cohort and the replication cohort, they're fairly comparable. And as I mentioned before, the majority of these individuals are of European ancestry, and I'll refer back to that in a moment. So the first thing we wanted to know is if there was an overall difference in the number of large rare CNVs observed in neuroblastoma patients versus controls. We did not see a difference. On average, there was 0.154 CNVs per case and 0.157 per control, with the p-value of 0.7. We did observe slightly more duplications than deletions in terms of these large rare CNVs. So ultimately, what we wanted to do was test individual copy number regions. So we looked for those that were greater than 500 kb and recurrently altered in at least 3 neuroblastoma cases. And this gave us a total of 40 CNVRs. We then used a Fisher exact test with a Bonferroni corrected significance threshold based on the number of copy number variable regions we were testing. Once we did that, there was only one region that passed genome-wide significance, and that was, that was a deletion on chromosome 16p11.2. It was present in 3.36% of cases and 0.02% of controls in the discovery cohort, similar levels and frequency in the replication cohort. This was highly significant and had an odds ratio of 13.9, which is again much higher than what one would typically see from a common variant identified through GWAS, which is more in the 1.1 to 1.2 or maybe up to 1.5 level. So this was mixed ethnicities. So we wanted to make sure that that wasn't confounding our results, because we know there is population stratification that we need to contend with. So therefore, we restricted to European only. And again, this remained highly significant. The frequencies were comparable, the p-values comparable, and the odds ratio, again, is comparable. So it turns out that this corresponds to a known microdeletion syndrome at 16p11.2. The deletion is known to affect about 1 in 3,000 individuals. It's been associated with multiple and diverse phenotypes, most notably autism spectrum disorder, but also developmental delay, intellectual disability, obesity, and others. When we look at the region that we identified, we can see, so what we're doing here is plotting the SNP array minimal and maximal regions. This was also confirmed by whole genome sequencing. And when we compare it to known published regions and the critical region, our data is very consistent with that. And as noted by us and others, this region is flanked by segmental duplications, and this is probably what is driving the copy number variation. It's a complicated region to study. It contains 28 protein coding genes. So this has hindered and complicated efforts to understand what may be a causal gene at the locus. We wanted to know, if we looked at matched tumor DNA, whether we could, one, confirm that the deletion was also present, and also to see if there may be a homozygous deletion or a deletion of the one remaining allele in the tumor. We did not find any homozygous deletions, but we did confirm the deletion using whole genome sequencing in all tumors, all germline samples that had a matched tumor. We next wanted to know if the deletion was enriched in any particular neuroblastoma subset or clinically relevant cohort. So we first looked at MYCN amplification status. Again, as I mentioned earlier, MYCN is a known neuroblastoma oncogene that associates with poor prognosis. And quite strikingly, we actually didn't see this deletion in any of our MYCN amplified patients with MYCN amplified tumors. So it definitely seems to be restricted to the non-amplified subset. And then similarly, we saw an enrichment really in the low risk, more benign form of the disease, although we did see it present in some high and also intermediate risk patients. So in many of these other phenotypes that have been shown to be associated with this microdeletion syndrome, oftentimes the deletion arises de novo instead of being inherited from one of the parents, although it can be inherited. So we really wanted to know, is this 16p11.2 microdeletion inherited or acquired de novo in the affected child with neuroblastoma? And this is where we really kind of hit a roadblock a while back, because there's a real limitation of current neuroblastoma studies that we do not have parental DNA. It's just not something that was routinely collected by the Children's Oncology Group, which banks our other samples. So this is where the Gabriella Miller Kids First Neuroblastoma X01 cohort really comes into play. So through our collaborator at UNC, Andy Olshan, who led the Neuroblastoma Epidemiology in North America, or NINA project, part of what they did was actually collect saliva DNA from patients and parents. And this is really what was the initial thrust for the X01 application. So we submitted approximately 640 cases, as well as their parents, with mostly saliva DNA. Some were supplemented from the biobank with blood DNA for sequencing. So this is really the first large study that includes parental DNA for neuroblastoma. And then we were fortunate enough to also be able to provide matched tumor DNA and RNA as indicated here, for 366 tumors with DNA and 226 with RNA sequencing, and these were banked through the Children's Oncology Group. And similar to the genome-wide association study, if you look at the clinical and biological characteristics at the right, these are really in line with the expected profile of the patient population. So we are very excited to start to utilize these data. So the very first question we wanted to ask was about the heritability of the 16p11.2 microdeletion. And we were fortunate enough that there were 4 cases that overlapped the cohort, the kids' first cohort. And what we're showing here are the 4, there are 2 trios and 2 duos on the bottom. And what we're plotting here is the coverage where you can see this is the maternal sample. This is the paternal sample. You can see that from this that there's no evidence for deletion in either parent. This is the child, which clearly has the deletion, so we infer that this is de novo. And similarly on the right, where we have both parents, we are able to say that the deletion in the child has occurred de novo. We can also further look at other low frequency variants, and we can say that this was inherited de novo on the paternal allele, whereas this deletion was inherited de novo on the maternal allele. So based on that limited data, we do not see a preference. We also had 2 individuals that had duo data available. So the child and only the mother was available through Kids First. And again, we were able to interrogate some low frequency variants, despite the fact that the paternal allele was not available, that we are fairly confident to infer that this is a de novo deletion that's inherited on the paternal allele. And similarly, although we didn't have paternal data available in this child either, we were able to infer that this occurred de novo on the maternal allele. So we think this is likely a de novo deletion that does not have preference for maternal or paternal alleles. So this is really our first look at the Kids First cohort. But I wanted to share with you, I realize this is very complicated, I'll try to walk you through it, but this is really our plan for the larger cohort. Some of the data I already mentioned is here in the middle, including the genome-wide association study, other data that we've generated in terms of epigenetic and 3D chromatin looping, as well as the Neuroblastoma Target Project. And our goal is to integrate that in with Kids First to do a primary analysis of the neuroblastoma triad and dyads. And again, we'll obviously be looking at whether mutations or rare variants are inherited or acquired de novo. We'll be looking for evidence of mosaicism. And again, we'll look at protein coding genes as well as non-coding regions by integrating in some of this epigenetic data and 3D looping data to understand if there are alterations in key regulatory regions or other non-coding regions such as non-coding RNA. We'll assess pathogenicity, enrichment testing, as well as other burden testing at pathway levels. And then ultimately, we're really interested in integrating the germline data with tumor data that is also being sequenced and provided through Kids First. So I mentioned that we have a large number of matched tumors that have undergone DNA whole genome sequencing or exome sequencing, as well as RNA sequencing. So we'll be looking for second hits, as well as other somatic mutations, looking at mutational signatures, doing eQTL analysis, fusion analysis, and so on. And in terms of the germline genetics, we're also in the process of performing a pan-childhood cancer data integration with these, with the neuroblastoma data. And a lot of this has been facilitated through the Kids First Data Resource Center, which is really enabling all of this large-scale work to be done and integrated both across cancer phenotypes and structural birth defects. So in summary of what I presented today, familial neuroblastoma is very rare. We know these are caused by mutations in ALK or FOX2B. We also know that there's incomplete penetrance and there's an autosomal dominant inheritance pattern. And the search for genetic modifiers and additional familial neuroblastoma predisposition genes is ongoing. A large effort is focused on the underlying drivers of sporadic neuroblastoma, which we think is a complex genetic disease. We know that common variants contribute to susceptibility. We know that through our GWAS studies. We also know that the GWAS has pointed us to unbiased identification of genes that drive both initiation and maintenance of the malignant phenotype. And therefore, there's a lot of potential translational opportunity there. We also know that there's rare pathogenic germline variants, such as in BARD1, that are enriched in neuroblastoma. And these have much larger effect sizes than what we see through the common variants from the GWAS. And by integrating the existing SNP array data with some of the Kids First data, we've identified a de novo rare germline 16p microdeletion that's associated with neuroblastoma. So this really adds to a very diverse array of phenotypes that this deletion syndrome is associated with, and really the first report of an association with cancer. So we're looking now to extend this to other inherited and acquired de novo mutations in neuroblastoma through the Kids First cohort. And we're really excited to explore the common and rare variants, the interaction of those, as well as the interaction between the host and tumor genome. And finally, we'll be intersecting these data with other cancer types, as well as structural birth defects cohorts through Kids First. And ultimately, I think this data really underscores, in terms of understanding the rare variants that drive childhood cancer, that large sequencing studies will be necessary. And with that, I would like to acknowledge the many people and groups that have contributed to this, as well as our funding supporters. And thank you very much.